Okay, 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 okay.
Good morning. Can you hear now?
Yes, ma'am.
Yes, ma'am.
Yes,
Well,
ma'am.
So today's class is on cardiac muscle. So majority of the details about the cardiac muscle you have studied along with the nerve muscle physiology. So we will be going to the details which are needed in the cardiovascular system only. So cardiac muscle, you know, the fibers are striated muscles and cardiac muscle is an involuntary muscle. Fibers are striated and muscle is involuntary, and each muscle fiber has a central nucleus. Okay, so you know the cardiac muscle fibers, they branch and connect with branches of other fibers. And at the point of contact between two cardiac muscle fibers, the membrane of both fibers will be showing extensive series of folds. So that is called as intercalated disc. Okay, so the muscle cardiac branching cardiac muscle fibers, they are connected to each other by this intercalated disc. So this is the region where the membranes of both the muscle fibers which are in contact, they show extensive series of folds. These intercalated discs, they are important during contraction of cardiac muscle, and it has two components. First one is desmosomes. Second is the gap junctions. Intercalated disc is having two components. First one is the desmosome. Second is the gap junctions. So desmosomes, they are strong connections that tie adjacent cells together. Desmosomes are strong connections that tie adjacent cells together. So what happens is, pull on one contractile unit can be transmitted to the next cell. So these desmosomes are important in the contraction of cardiac muscle. They are strong connections that tie adjacent cells together so that pull on one contractile unit can be transmitted to next cells. Then there is gap junctions. Gap junctions, they are providing low resistant bridges. These gap junctions, they electrically connect cardiac muscle cells to one another. So intercalated disc is having two components, desmosomes and the gap junctions. So desmosomes are connections which are leading to the contraction of the cardiac muscle fibers, and gap junctions are the connections which are needed for electrical connectivity of cardiac muscle cells. So gap junctions, they provide a low resistant bridge that electrically connect cardiac muscle cells to one another. So they allow wave of depolarization to spread rapidly from cell to cell. Gap junctions allow the spread of depolarization rapidly from cell to cell so that the cardiac muscle act as a single unit. Okay, they are contracting simultaneously. So the cardiac muscle is said to be a functional syncytium. Okay, gap junctions, they are actually providing low resistant bridges that electrically connect cardiac cells to one another, cardiac muscle cells to one another. So it allows the wave of depolarization to spread rapidly from one cardiac muscle cell to other, so that cardiac muscle contract simultaneously. It act as a single unit, so that the cardiac muscle is called as the functional syncytium. Okay, in this diagram there is it is shown the spiral arrangement of ventricular muscle. Okay, ventricular muscle is arranged in this specific way, so that the ventricular contraction can skews the blood upward from the apex of the heart. So that's just diagrammatic representation of how these muscles are arranged. Now we will see what are the difference between the cardiac muscle and skeletal muscle. About the location of T tubules, cardiac muscle it is at the said line, and skeletal muscle it is at the air junction. About the diameter of the T tubules, it is more in cardiac muscle. Narrow tubular cisternae in case of cardiac muscle, and large dilated cistern in case of skeletal muscle. Then regarding the association of T tubule, dyad, one tubule and one cistern in case of cardiac muscle, and triad, one tubule and two cistern in case of skeletal muscle. And about the sarcomeric organization, it is more regular in the skeletal muscle. It is not so well developed in the cardiac muscle. So these are the differences between the cardiac and skeletal muscle. So you may be knowing all this. Now we'll go to what are the different types of cardiac muscle. Okay, so cardiac muscle, it can be divided into three types. First, that is the contractile muscle fibers. These muscle fibers, they form bulk of atria and ventricle. Okay, they are for contraction purpose. Second, there is nodal tissues. That is the SA node and AV node. Okay, they contain pacemaker cells and they can produce their own rhythm. And the third type is conducting tissues. So that include bundle of his Purkinje fibers and atrial internodal fibers. Okay, so these conducting fibers, they are actually specialized excitatory and conducting fibers. They contract only feebly. Okay, contraction is mainly by the atrial and ventricular muscles. Conducting tissues contract only feebly. Okay, they contain few contractile fibrils. Their main role is in showing autorhythmicity or conduction. Okay, so this is important aspect of cardiac muscle. It is having three types of muscle: contractile muscle fibers, nodal fibers, and the conducting tissues. Now about the conducting system. So they are actually first about the contractile muscle fibers. Contractile muscle fibers having the same features as that of skeletal muscle. They are helping in the contraction of cardiac muscle. So they are almost similar to the skeletal muscle. Then about the conducting fibers, they are modified cardiac muscles. They have only few striations, and the boundaries of these muscle fibers are indistinct. Then SA node and AV node, they contain cells called as pacemaker cells or P cells. Okay, hope it's clear. Now we'll see what are the properties of cardiac muscle. So electrical properties, what are the properties coming under electrical properties? First one is called as the excitability. It is having other name, that is bathmotropic effect. Second, autorhythmicity. It is also called as chronotropic effect. Third feature, that is conductivity, that is called dromotropic effect. Okay, so these are the main properties coming under electrical properties: excitability, autorhythmicity, and conductivity. Then, property coming under mechanical property, that is contractibility. It is also called as inotropic effect. So these are the properties of main properties of cardiac muscle: excitability, autorhythmicity, conductivity, and contractility. Okay, hope you are following.
Yes, ma'am.
Now
Yes.
we will see each of these properties. What is excitability?
Ma'am, can you show the previous slides just once?
What is excitability? Anyone?
Response to stimulus.
What is it? What do you mean by an excitable tissue? What are the excitable tissues?
Neurons and
Nerves.
muscle fibers.
Okay, neurons and muscle fibers, they are the excitable tissue. What is excitability?
They undergo
Response to
depolarization and response to stimulus.
Excitability is the ability to respond to a stimulus. Okay, excitability means it is the ability to respond to a stimulus of adequate strength and duration. Okay, the stimulus should be of adequate strength and duration. So the ability to respond to a stimulus of adequate strength and duration is called as excitability. So here in the cardiac conducting fibers, it is by generating a propagated action potential. Okay, ability of the stimulus by generating a propagated action potential, that is start happening here. Then what is a stimulus? What is a stimulus?
it can be mechanical, electrical, or chemical.
Okay, what is it?
Any anything that elicitate a response.
Okay, so it is any event or any force which, when applied to an excitable tissue, can produce a characteristic response. Okay, that is stimulus. Any event or force when applied to an excitable tissue producing a characteristic response, that is a stimulus. So excitability, that is the ability to respond to a stimulus. Okay, now there are ion channels which can be divided into voltage gated and ligand gated ion channels. So these ion channels are involved in functioning of the cardiac muscle action potential. So main role is for the voltage gated channels, which include sodium channel, calcium channel, and potassium channel. Okay, so we will discuss the more details along with the steps of the action potential. So main channels involved in the action potential, cardiac muscle action potential is these channels: sodium, calcium and potassium. Before going to action potential, I want to clear some terms. What is polarization? What do you mean by the term polarization? That is any state when the membrane potential is other than 0 millivolt. That is called as polarization. Okay. Then what is depolarization?
The reversal of polarity on
Okay.
application of stimulus.
Okay. Decrease in the potential than the resting membrane potential. So that is depolarization. Then repolarization. That is membrane returns to resting potential after depolarization. That is meant by repolarization. The next term is hyperpolarization. So membrane becomes more polarized than the resting potentials. So these are the four terms that you know. What is polarization, what is depolarization, repolarization and hyperpolarization. So all these concepts you have covered in along with membrane potential, I think. So hyperpolarization, that is increase in potential. Then there is one more term, that is overshoot. When inside of the cell becomes positive due to reversal of membrane polarity, that is called as overshoot. Okay, hope these terms are clear to you. So we are using these terms when we are describing the action potential. So coming to the electrical potential of cardiac muscle. Cardiac muscle resting membrane potential is minus 85 to minus 95 millivolt. Okay, negative interior with reference to exterior. This is the resting membrane potential of the contractile cardiac muscles. That is the muscles of the atria and ventricle. Okay, now coming to action potential. So what is action potential?
Stimulus causing potential change greater than the threshold value.
Okay, action potential can be explained as there is a rapid change in the membrane potential followed by returning to normal resting level. Okay, so there will be a reversal of the membrane polarity followed by restoration of the normal polarity. So that is action potential. In simple terms, that is rapid change in the membrane potential followed by returning to resting level. In cardiac muscle, it is divided into 5 distinct phases. Okay, so these phases are phase 0. First phase, phase 0, that is rapid depolarization. So you can look at the diagram here. So this phase is the phase 0. That is the phase showing rapid depolarization. Second stage is phase 1, that is initial rapid repolarization. Third is the phase 2, that is the plateau phase. Next phase is phase 3, that is the repolarization phase. Next phase is the phase 4 or the resting potential. So these are the phases of action potential of cardiac muscle. Okay, phase 0, rapid depolarization. Phase 1, initial rapid repolarization. Then there is a plateau phase. Then phase 3 is repolarization, and phase 4, that is resting potential. So hope the phases are clear to you. Cardiac muscle action potential are divided into 5 distinct phases: phase 0, phase 1, phase 2, phase 3, and phase 4. Now we'll see each of them. First one, that is the phase 0, that is rapid depolarization. Okay, so we have already explained the resting membrane potential. It is between minus 85 to minus 95 millivolt. Generally, we consider it as minus 90 millivolt. So when the cardiac muscle is stimulated, there will be production of the action potential. So what happens in the first phase? So when the cardiac muscle fibers are activated, the first stage is depolarization. It proceeds rapidly. Depolarization, which proceeds rapidly, there is an overshoot. Okay, now overshoot is present. That is in as in skeletal muscle action potential and nerve action potential. Here also there is an overshoot. Okay, the amplitude of potential may reach up to plus 20 to plus 30 millivolts. So we are starting from a resting membrane potential of minus 85 to minus 95 millivolts. After depolarization, that potential may go up to plus 20 or plus 30. And the phase 0, that is a rapid depolarization phase, is only for 2 milliseconds. Okay, so that is the phase of rapid depolarization. Hope it is clear.
Yes, ma'am.
Now, what is the ionic basis behind this phase? What are the ionic bases behind this phase? So first, there are two bases for this phase. That is, two things are happening. First, there is rapid opening of voltage gated sodium channels. Okay, first there will be rapid opening of voltage gated sodium channels, and there will be rapid influx of sodium ions. Voltage gated sodium channels get opened and sodium enters into the cardiac muscle fiber. So that is the first thing. After the opening of the sodium channels, the resting membrane potential, when it reaches the level of minus 30 to minus 40 millivolts, then the calcium channels get open up and there will be influx of calcium ions also. Okay, so there are two channels which are operating in phase 0. First, there will be depolarization. First, there will be depolarization due to rapid opening of the sodium channels, resulting in influx of sodium ions. Then, when the membrane potential reaches to minus 30 to minus 40 millivolts, calcium channels open up. And there will be influx of calcium ions also. So these two are contributing to the first phase, that is the phase 0. Okay, so rapid depolarization, the membrane potential from minus 80 to minus 90, it will reach up to plus 20 millivolts. Then next phase is phase 1, that is initial rapid repolarization. So as the term, it will be for short duration. Rapid depolarization is followed by very short lived slight rapid repolarization. Okay, here the membrane potential reverses its polarity, and from this plus 20 to plus 30 millivolts, it can come to minus 10 millivolt during this phase. Okay, it can come up to minus 10 millivolts. Okay, so what is the ionic basis behind this? Initial rapid repolarization is due to inactivation of sodium channels. First, there will be inactivation of sodium channels. Then, there will be opening of potassium channels. There is a channel which is named as transient outward potassium channels. So, these channels get opened. So, there will be resulting transient outward current. That is potassium efflux. Potassium is going out. Okay, the phase 1 initial rapid repolarization, that is due to two things. First, there is inactivation of sodium channels. So the sodium influx stops. Second is efflux of potassium ions by opening of transient outward potassium channels that result in potassium efflux. So the sodium influx stopped and potassium efflux has begun. Potassium is going out of the cell. Okay. Next phase is phase 2, that is the Plato phase. Okay. So this Plato phase, what is happening? Membrane repolarize very slowly. Okay. So the membrane potential falls very slowly. So it will fall only to minus 40 milli. It can fall up to minus 40 millivolt during this phase. And this Plato phase, it can last for about 100 to 200 milliseconds. So the phase 2, Plato phase, here the membrane potential repolarizes very slowly, very slowly. And the potential can go up to minus 40 millivolts in this phase. And this phase is a longer phase and it lasts for about 100 to 200 milliseconds. Okay, so what will be the ionic basis behind this? Here, very slow repolarization is due to first one, slower but prolonged opening of voltage gated calcium channels. Okay, so these calcium channels are L-type calcium channels. Calcium channels can be divided into two types, that is transient channels and long-lasting channels. So this L-type means they are long-lasting calcium channels. Okay, this channel promote slower influx of calcium. Okay, when the L type or long lasting type of calcium channels get opened, they allow slow inward movement of sodium. That is the first point. Second point, there will be closure of a distinct set of potassium channels. Okay, that is called as inward rectifying potassium channels. They will be closed. Okay, actually, potassium channels normally open when the membrane depolarizes. But in cardiac muscle, this type of specialized type of inward rectifying potassium channels, they are closing. Okay, during depolarization, they are closing. So the ionic basis behind the phase Plato phase is that there are two things. First, there is opening of L type calcium channels which allows slow entry of calcium. Second is closure of a particular type of potassium channels. It is called as inward rectifying potassium channels. Okay, so that's about phase 2. And next phase is phase 3, that is repolarization. So here, complete repolarization occurs and the membrane potential falls to approximate resting value. And this phase lasts for about 50 milliseconds. Okay, so what is happening in phase 3? For complete repolarization, what is happening? It results from closing of the calcium channels. Okay, the calcium channels which are opened, that will be closed. Second thing is opening of two types of potassium channels. So that will be leading to delayed increase in potassium efflux. So two channels are there. Delayed outward rectifying potassium channels. Okay. First type of potassium channel which are opened during this phase is delayed outward rectifying potassium channels. Okay, they are promoting potassium efflux. Second type of potassium channels, they are calcium activated channels which are activated by elevated sarcoplasmic calcium levels. So there are two types of potassium channels: delayed outward rectifying potassium channel and calcium activated channels. Both will be opened so that there is potassium efflux. Okay. And coming to the last phase, that is resting potential. Okay, so that resting potential is maintained at minus 90 millivolt. So this is maintained by a resting potassium current. Are you listening?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
So these are the phases of the cardiac muscle action potential: phase 0, phase 1, phase 2, phase 3, and phase 4. And ionic basis of each channel. Hope it is clear to you. Okay, the resting ionic composition is restored by sodium potassium ATPase pump also. Okay. So here the duration of action potential
Ma'am,
is
can you show the
about
previous slide, please?
1 minute. Duration of action potential is about 250 milliseconds. Okay, when the heart rate is 75 per minute, the duration of action potential of cardiac muscle, it will be around 250 milliseconds. What is the duration of action potential in a nerve? What is the
0.1
duration of action potential?
0.1
How
second.
much? How much? How much is the action potential of nerve duration?
0.1 second.
Okay, 1 millisecond. Okay, so that's about the action potential in cardiac muscle. So these are the stages. Okay, phase 0, you can see there is mainly there is sodium influx, and phase 1 there is, yes.
Can you show the previous slide?
1 minute, this is the last slide. So phase 1, there will be, what is happening during phase 1? Sodium channels are inactivated and potassium channels are get open. So potassium efflux is there. Second, phase 2, there is influx of calcium, slow influx of calcium and outflux of potassium. Then there is phase 3. So there is potassium efflux by the channels. That is mainly by 2 channels. Then the fourth, it coming back to resting membrane potential. So these are the phases of cardiac action potential. So you should study this well. Okay. So we will stop today's class here. The rest we will continue in the next class. So all of you read today's topic. Okay. Next class is based on this. Today we have seen the cardiac muscle action potential, that is mainly of the contracting fibers. Okay, next day we will be seeing the action potential of conductive fibers of the heart. Okay, thank you.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you, 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 ma'am.
Yes. Hello. Yes.
Good morning, ma'am.
Good morning. Yes. Today we will start on Sari. As hope everybody is listening. എല്ലാരും കേട്ടോണ്ടിരിക്കുകയെന്നെ?
Ma'am, can we have a
Yes, ma'am.
5-minute break?
You want a break?
Yes, ma'am.
5 minutes break, huh? Take
Yes,
5
ma'am.
minutes. 5
Thank
minutes.
you, ma'am.
Okay, okay. Yes, can we start? എല്ലാരും തിരിച്ചെത്തിയോ? Yes. Yes. So last class we were dealing with glycine metabolism. Today we are moving on to serine metabolism. Yes. What do you know about serine? Yes. What do you know about serine? Serine? It's a yes. Yes. Serine, you know, it is a hydroxy amino acid. No, serine, it is a hydroxy amino acid. It is a non-essential amino acid. Understand? Serine is a hydroxy amino acid. It is a non-essential amino acid, and also it is glucogenic. Serine is glucogenic. Understand? How it is serine synthesized? How is serine synthesized? What is the step? From which amino acid serine is being synthesized?
Glycine.
Yes, serine is synthesized from glycine. How? With the help of hydroxy methyl transferase enzyme and accepting a one carbon group from one carbon pool. The enzyme is serine hydroxy methyl transferase forming serine. Understand? From methylene THFA. N5N10 methylene THFA. It will accept a methyl group forming serine. And it is a freely reversible reaction. The major contributor of serine is phosphoglycerate. Phosphoglycerate is a major contributor of serine. Understand? So the major contributor of serine is phosphoglycerate. Phosphoglycerate will undergo oxidation forming phosphopyruvate. Forming phosphohydroxypyruvate. Phosphohydroxypyruvate will undergo oxidation in presence of a hydrogenation reaction forming phosphohydroxypyruvate. This phosphohydroxypyruvate will undergo a transamination reaction. Phosphohydroxypyruvate will undergo a transamination reaction forming phosphoserine. Understand? Forming phosphoserine. And this phosphoserine is hydrolyzed in presence of phosphatase enzyme and generates serine. So the major contributor of serine is phosphoglycerate. Phosphoglycerate undergoes oxidation forming phosphohydroxypyruvate, and this phosphohydroxypyruvate will undergo a transamination reaction forming phosphoserine, and from phosphoserine will undergo hydrolysis by removing the phosphate group forming serine. So the major contributor of serine is from phosphoglycerate. Another contributor is alanine. It can be synthesized from alanine. How it can be synthesized from alanine? It can be synthesized from alanine by a transamination reaction. Alanine in presence of hydroxypyruvate. Alanine reacts with hydroxypyruvate forming serine and pyruvate. Forming serine and pyruvate. Understand? That is a transamination reaction. So coming to the synthesis of serine, serine is synthesized from glycine, from phosphoglycerate, and also from alanine. Understand? That is the synthesis of serine. Now coming to the biological role of serine. What are the biological role of serine? Biological role of serine. What are the biological role of serine? Yes. What are the uses of serine? Serine is a major one carbon donor. Understand? Serine is a major one carbon donor that you have studied in the last class. So serine is an important one carbon donor for the synthesis of glycine. And also it is used for the synthesis of glycine. Understand? From serine, glycine can be synthesized. So it is a major one carbon compound and it is used for the synthesis of glycine. And also from serine, choline can be synthesized. From serine, choline can be synthesized. What is choline? Choline is trimethyl ethanolamine. Trimethyl ethanolamine. How from choline? How from serine choline is synthesized? Serine will undergo a decarboxylation. From serine, a carboxyl group is removed as carbon dioxide by a decarboxylase enzyme forming ethanolamine. Forming ethanolamine. So what is the structure is? CH2 NH2. So that is ethanolamine. Understand? This ethanolamine. So this is the structure of ethanolamine. Ethanolamine will accept 3 methyl group from 1 carbon. Understand? This ethanolamine will accept 3 1 carbon group from 1 carbon pool. How? See? There is CH2. 3 1 carbon group from 1 carbon pool forming choline. Understand? Serine will accept 3 1 carbon group from 1 carbon pool forming trimethyl ethanolamine. And this trimethyl ethanolamine is choline. So the important role of serine is it is used, it's an important 1 carbon donor. It is used for the synthesis of glycine. It is used for the synthesis of choline. How choline is being synthesized? Serine will undergo decarboxylation forming ethanolamine, and ethanolamine gets methylated forming trimethyl ethanolamine, that is choline. And from this choline, we can generate glycine, and from glycine again, we can generate serine. Understand? So that is a vicious cycle. And other important roles of serine are, it is used for the synthesis of glycolipids. Serine is used for the synthesis of glycolipid. It is used for the synthesis of phospholipid. It is used for the synthesis of phospholipid. And it's also an important component of protein. Serine is also an important component of protein. And also another important function of serine is that serine is needed for the synthesis of the 21st amino acid, that is selenocysteine. Selenocysteine is a 21st amino acid. So serine is needed for the synthesis of the 21st amino acid selenocysteine. And this was discovered in the year 1986. Which is the 22nd amino acid? Yes, Surya. Which is the 22nd amino acid? Anybody? Advait? Yes. Please.
Hydroxyproline.
Yes, pyrrolysine. No. Hydroxyproline, not hydroxyproline. That is pyrrolysine. Amino acid classification at the padichillarno. Eh? So it is pyrrolysine. So the 21st amino acid is selenocysteine and the 22nd amino acid is pyrrolysine. Now, we are not going to the details how selenocysteine is being synthesized. I will just touch upon the major points only, how selenocysteine is being formed. So, coming to selenocysteine. Selenocysteine is an important component in. Selenocysteine is mainly present in 3 enzymes. One is thioredoxin reductase. Thioredoxin reductase. Thioredoxin reductase. What is thioredoxin reductase? Then deiodinase. And third one is glutathione peroxidase. Glutathione peroxidase. These are the 3 major enzymes containing glutathione peroxidase. These are the three major enzymes containing selenocysteine. What is the use of thioredoxin reductase? Thioredoxin reductase is used for the synthesis of deoxyribonucleotide. Thioredoxin reductase is used for the synthesis of deoxyribonucleotide. And what is the use of deiodinase? Deiodinase is used for the conversion of T4 to T3. Conversion of T4 to T3. Understand? T4 to T3. And what is the use of glutathione peroxidase? It is used for scavenging free radicals. So selenocysteine is mainly present in three enzymes, that is thioredoxin reductase, which is used for the synthesis of deoxyribonucleotide, deiodinase for the conversion of T4 to T3, and glutathione peroxidase, it is used for scavenging free radicals. And we will see how the major points how selenocysteine is being formed. Selenocysteine, the carbon skeleton of selenocysteine is being derived from serine. The carbon skeleton of selenocysteine is being derived from serine. Understand? So the carbon skeleton is derived from serine, and the selenium is being provided by selenophosphate. Selenophosphate, which is being synthesized from a molecule of ATP and selenate. Understand? And a tRNA, a specific tRNA is needed for the synthesis of selenocysteine. So selenocysteine, for the synthesis of selenocysteine, serine, selenophosphate, and a specific tRNA is being needed. Serine contributes the carbon skeleton, whereas the selenophosphate will provide the selenium ion, and tRNA will carry the amino acid. Say in the protein synthetic pathway, so there is no specific codon for the formation of selenocysteine. The UGA codon. What is UGA codon? Normally UGA codon is a stop codon. But in selenocysteine formation, it is acting as a codon for the synthesis of selenocysteine. Understand? There is no specific codon for selenocysteine formation, but there is a specific tRNA for it. The tRNA will carry serine. The tRNA will carry serine to the active site, and this serine is being converted to selenocysteine by a specific insertion element, that is selenocysteine insertion element. The serine is being converted to selenocysteine. Understand? This is the major reaction how selenocysteine is getting formed. Understand? Major reaction how selenocysteine is getting formed. Now coming to the structure, I didn't show you the structure of selenocysteine. It has a structure very similar to cysteine itself. The selenocysteine has a structure very similar to cysteine itself. See? So this is the structure of cysteine. Now coming to the structure of selenocysteine. Only difference is the sulfur is being replaced by selene. Understand? It is being replaced by selene. So this is the structure of selenocysteine. Similar to cysteine, only the sulfur is being replaced by selene. Understand? So the major role of, what are the major role of serine? It's an important one carbon donor. It is used for the synthesis of choline. It is used for the synthesis of glycine. And it's an important component of phospholipid, glycolipid, and important for the formation of protein and also for the synthesis of selenocysteine. Understand? These are the major points under serine. Now, moving on to alanine. Alanine. I think alanine, again, you know, alanine is a glucogenic amino acid. It's a glucogenic amino acid. And how it is formed? How is alanine formed?
From
Yes.
pyruvate.
Yes, from pyruvate and glutamate, which is the enzyme alanine transaminase. By transamination reaction, alanine is getting formed by glutamate plus pyruvate in presence of ALT, alanine transaminase enzyme. It forms alanine by a transamination reaction. And you have studied this glucose alanine cycle, Cahill cycle. What is the importance of Cahill cycle? What is the importance of Cahill cycle or glucose alanine cycle? It was dealt along with urea cycle. Yes.
Alanine transported to the liver.
Yes, alanine is being transported to liver for the detoxification of ammonia and also to maintain blood sugar level. Cahill cycle mainly operates to maintain blood sugar level. Understand? And another thing that you haven't dealt with is beta alanine. Beta alanine. What is beta alanine? In beta alanine, it is different from alanine is that yes. In beta alanine, the amino group is attached to the alpha carbon. Amino group is attached to beta carbon. Understand? That is beta alanine. And beta alanine is being formed as a end product in pyrimidine metabolism. In pyrimidine catabolism, beta alanine is getting formed. And what is the use of beta alanine? Beta alanine is used for the synthesis of coenzyme. Understand? So alanine, I'm not going into the details of alanine. You can read from the text. Alanine, it's a glucogenic amino acid. It is synthesized from pyruvate by transamination reaction. And important one is glucose alanine cycle and also beta alanine. Beta alanine is an intermediate, is an end product in pyrimidine catabolism, and beta alanine is needed for the synthesis of coenzyme A. That much about alanine. And we are moving on to an important topic, that is sulfur containing amino acid. Sulfur containing amino acid. Which are the sulfur containing amino acid?
Methionine.
Methionine and cysteine. Important, usually asked as an essay. Just similar to glycine, the sulfur containing amino acids. Important amino acid metabolism important glycine, then methionine, cysteine, sulfur containing amino acids, then aromatic amino acids. Which are the aromatic amino acids? Phenylalanine, tyrosine, tryptophan, then histidine. This is important essay. Others can be asked as short notes only. Now, coming to the sulfur containing amino. Coming to the sulfur containing amino acids. Sulfur containing amino acids. See, mainly this organic sulfur is derived in our body, in human body. This organic sulfur is mainly derived from sulfur containing amino acids, sulfatides, and also glycosaminoglycans. Understand? Organic sulfur in human body is derived mainly from the sulfur containing amino acids, glycosaminoglycans, and sulfatides. And the major dietary product of sulfur is methionine. Now today we will deal with methionine metabolism. Methionine. You know what is methionine? Methionine, again, what is the specialty of methionine? Methionine is an essential amino acid. Yes, methionine is an essential amino acid. It is a sulfur containing amino acid. It is a sulfur containing amino acid, and also it is glucogenic. Methionine is glucogenic. From methionine is catabolized to succinyl-CoA, from which glucose can be synthesized. So methionine is glucogenic. What is the special 3-letter abbreviation for methionine? 3-letter abbreviation is MET,
MET.
and the
MET.
single letter abbreviation is M. Understand? So coming to the structure of methionine, COH, CH, NH2, CH2, CH2, S, CH3. Understand? So this is the structure of methionine. So here the methionine, the sulfur is seen as a sulfur is in thioether linkage. Understand? In methionine, the sulfur is in thioether linkage. Sulfur test പഠിച്ചായിരുന്നോ? Color reactions പഠിപ്പിച്ചായിരുന്നോ? Along with amino acid metabolism, amino acid along with color reactions എടുത്തിട്ടുണ്ടായിരുന്നോ? Along with this chemistry, amino acid chemistry എടുത്തപ്പം color reactions എടുത്തോ? Yes, Vidhu. Naji. കുട്ടികളെ respond ചെയ്യൂ. Yes.
I don't think so.
Don't think so. Yes. See, color reactions in the sulfur test. Sulfur test will answer the reactions of sulfur containing amino acid. Only cysteine is being answered. Methionine can't be answered since the sulfur is in thioether linkage. Since sulfur is in thioether linkage, sulfur test is in being answered. But cysteine will answer sulfur test. I think practical we will deal with color reactions. So see, in methionine, the sulfur is seen in thioether linkage. So what is methionine? Methionine is an essential amino acid. It is a sulfur containing amino acid. It is glucogenic. Now coming to the major biological role of methionine. Methionine is an important methyl donor, the active form of methionine. Creatine പഠിപ്പിച്ചപ്പോൾ പഠിച്ചു. The active form of methionine is S-adenosyl methionine. It is the active form of methionine and it is used for it is an important methyl donor. So today we'll deal with how from methionine, the active methionine is being synthesized. How from methionine, the active methionine is being synthesized. Yes. How from methionine, the active yes. How from methionine, the active methionine is being synthesized. Here, methionine in presence of adenosyl methionine transferase enzyme utilizing a molecule of ATP. Here, a molecule of ATP is being utilized. From ATP, the adenosyl group is transferred to methionine, forming S-adenosyl methionine. And the 3 high energy phosphate is being removed as 2 molecules of pyrophosphate and 1 inorganic phosphate. Understand? All of the high energy phosphate is being removed from ATP. The only remaining is adenosyl group, which is transferred to methionine forming, which is then transferred to methionine. So it is being transferred to methionine forming S-adenosyl methionine. CH2, CH2. I think it's better you write along with me. See, the adenosyl group of ATP. Adenosyl group. And now the methyl group is being attached by a labile bond. Understand? So this is S-adenosyl methionine. From ATP, the adenosyl group is attached to the sulfur, and now the methyl group is in a labile bond, attached by a labile bond. So it can be easily be removed. So this is the structure of S-adenosyl methionine. The enzyme is adenosyl methionine adenosyl transferase enzyme. The enzyme is methionine adenosyl transferase. Understand? So methionine in presence of adenosyl transferase, the adenosyl group is attached to methionine, forming S-adenosyl methionine. Now the methyl group is attached by a labile bond. So it can be easily be removed. Now this S-adenosyl methionine in presence of an acceptor, methyl acceptor. The methyl acceptor will accept the methyl group, forming methyl acceptor, and itself is converted to, so the methyl group is removed. Understand? Forming H2, CH2, CH2. So the methyl group is removed, forming understand? This is S-adenosyl homocysteine. You have studied one methylation reaction. Which was that? Last class you dealt with a methylation reaction in the synthesis of creatine, where S-adenosyl methionine is acting as a methyl donor, which is a substrate that is accepting methyl group. S. Please students, you respond. S.
Guanidoacetate.
Yes, guanidoacetate will accept a methyl group from S-adenosyl methionine, forming creatine. Understand? So that is an example for a transmethylation reaction where S-adenosyl methionine is the methyl donor. This is a reaction referred as transmethylation reaction. Transmethylation reaction. The enzyme is methyl transferase. Understand? So the acceptor will accept a methyl group forming methyl acceptor, and the remaining one is S-adenosyl homocysteine. And this, now before going to that, we will deal with the transmethylation reaction. We'll deal with transmethylation reaction. I will just discuss on transmethylation reaction and then move on to the further steps of methionine metabolism. Transmethylation reaction is very important, usually asked as a short note for you. Transmethylation reaction. Transmethylation reaction. See, in transmethylation reaction, for transmethylation reaction, the methyl donor is S-adenosyl methionine. S-adenosyl methionine is the methyl donor. Which are the reactions? Transmethylation reaction. One is guanidoacetate to creatine. Guanidoacetate to creatine. Understand? Guanidoacetate to creatine is one transmethylation reaction. The another is norepinephrine to epinephrine. Norepinephrine to epinephrine. Norepinephrine to epinephrine. Norepinephrine is methylated to epinephrine. Then third one is epinephrine to metanephrine. Epinephrine is methylated to metanephrine. Metanephrine. And another one is tRNA. Will undergo methylated tRNA to form methylated tRNA. Methylated tRNA. And another important one is acetyl serotonin. Formation of acetyl serotonin. Serotonin to melatonin. This all reaction you will come along with this norepinephrine, epinephrine metabolism will come along with phenylalanine metabolism and acetyl serotonin along with tryptophan metabolism. So when transmethylation is being asked as a short note, you have to give what is transmethylation reaction? Transmethylation reaction is the reaction in which the methyl donor is S-adenosyl methionine, and it is used for the synthesis of creatine, compounds like creatine, epinephrine, metanephrine, methylated tRNA, and for the synthesis of melatonin. And the enzyme is methyl transferase enzyme. Transmethylation reaction is very important, usually asked as a short note for you. Is it clear? When the methyl group is donated to a methyl acceptor, the S-adenosyl methionine is converted to S-adenosyl homocysteine. Is it clear, students? Yes, please do respond.
Yes, ma'am.
Yes.
Yes. Coming to methionine metabolism. Methionine, it's an essential amino acid. It is a glucogenic amino acid. It is a sulfur-containing amino acid. And it is used mainly for the synthesis of the active methionine, that is S-adenosyl methionine. Methionine will undergo first methionine in presence of adenosyl transferase enzyme, methionine in presence of ATP. The 3 high energy phosphate is being removed, and the adenosyl group is attached to the methyl group to the sulfur atom of methionine, forming S-adenosyl methionine. And this S-adenosyl methionine will donate the methyl group to its acceptors during transmethylation reaction and forms S-adenosyl homocysteine. I think it's clear up to these steps. Now, coming to the further metabolism. Yes. Now, this S-adenosyl homocysteine. What happens to S-adenosyl homocysteine? This S-adenosyl homocysteine in presence of an homocysteinase enzyme. Homocysteinase enzyme, the adenosyl group is removed. Adenosyl group is removed from S-adenosyl homocysteine. Understand? Adenosyl group is removed in presence of adenosyl homocysteinase enzyme. The adenosyl group is removed. Adenosyl group is removed. Adenosyl group is removed, forming CH2, CH2, SH. So what is this structure? When the adenosyl group is removed, it forms homocysteine. Understand? It forms homocysteine. Homocysteine. Understand? So methionine to S-adenosyl, methionine to S-adenosyl homocysteine, and in presence of an homocysteinase, adenosyl homocysteinase enzyme, adenosyl homocysteinase, in presence of adenosyl homocysteinase enzyme, the adenosyl group is removed, forming homocysteine. And coming to the, based on the cellular requirement, homocysteine methionine can be regenerated from homocysteine, or homocysteine can form cysteine. Understand? So based on the cellular requirement, homocysteine can either form methionine or can form cysteine. Understand? We will see how methionine is regenerated from homocysteine. How methionine is regenerated from homocysteine. Yes, from homocysteine, how methionine is regenerated. This homocysteine will accept a നമ്മൾ നേരത്തെ പഠിച്ചിട്ടുണ്ട് during 1-carbon metabolism. Methyl THFA, N5-methyl THFA. N5-methyl THFA is the most oxidized form of the 1-carbon group. N5-methyl THFA will donate a methyl group to cobalamin. Will give a methyl group to cobalamin forming methyl cobalamin. Forming methyl cobalamin. Understand? Forming methyl cobalamin. And this methyl cobalamin will donate a methyl group to homocysteine, forming methionine, regenerating methionine. Regenerating methionine. Yes. Is this step clear? Yes. Yes, students, please respond.
Yes,
Yes.
ma'am.
Yes. From homocysteine, a methyl group, N5-methyl THFA will donate a methyl group to vitamin B12, forming methyl cobalamin. And this methyl cobalamin donate the methyl group to homocysteine, forming methionine, regenerating methionine. Understand? Regenerating methionine. So this is methionine. Yes, is it clear? So from homocysteine, methionine can be regenerated. How? By accepting a methyl group from methyl cobalamin. The cobalamin is methylated by N5-methyl THFA. That I think we have dealt along with one carbon metabolism. So what happens in vitamin B12 deficiency? In vitamin B12 deficiency, this N5-methyl THFA will remain as N5-methyl THFA itself. Since the cobalamin can't be, since there is cobalamin deficiency, it can't be methylated. So it will remain as N5-methyl THFA itself, causing folic acid, that is folate trap. We dealt along with one carbon metabolism. You just recap it. Understand? So that is the importance of this step. So from homocysteine, methionine can be regenerated. Another pathway is formation of cysteine. Formation of cysteine. How? From homocysteine, cysteine is getting formed. That is why methionine is said to be essential amino acid. Can't be synthesized in our body. Methionine can't be synthesized. It only can be supplied through diet. It can't be synthesized. It is only regenerated from homocysteine. It is not synthesized from homocysteine. It is only regenerated from homocysteine. It can be synthesized from homocysteine, so methionine can be taken as a non-essential amino acid. No. Here, it is regenerated from homocysteine only. It is not synthesized from homocysteine. It is regenerated from homocysteine. Now, how from homocysteine, cysteine is getting formed. Cysteine is getting formed. This homocysteine will combine with serine. It's an important step. It will combine with serine. So this is the structure of serine. And the enzyme is cystathionin beta synthase. The enzyme is cystathionin beta synthase. It's an important enzyme. Cystathionin beta synthase. Cystathionin beta synthase. Beta synthase. It is a PLP dependent enzyme. Pyridoxal phosphate dependent enzyme. So which are the reactions requiring pyridoxal phosphate as a coenzyme? Pyridoxal phosphate is a coenzyme in transamination reaction, in decarboxylation reaction, and also for the synthesis of cysteine. That is, for cystathionin beta synthase, pyridoxal phosphate is acting as a coenzyme. So here, homocysteine will combine with serine in presence of cystathionin beta synthase enzyme, which is a pyridoxal phosphate dependent enzyme, by removing a molecule of water. By removing a molecule of water, forming cystathionin. Homocysteine will combine with serine in presence of cystathionine beta synthase enzyme, which is a PLP dependent enzyme forming cystathionine. Forming cystathionine. And a molecule of water is being removed, forming cystathionine. Structure on the data. Forming cystathione. See. Here comes ECH2 group. CH2, CH, COH, NH2. So this is the structure of cystathionine. Understand? Cystathionine in presence of cystathionine beta synthase. Cystathionine is synthesized from homocysteine, will combine with the serine, removing a molecule of water. The enzyme is cystathionine beta synthase. It is a PLP dependent enzyme forming cystathionine. And this cystathionine in presence of cystathionase enzyme. Cystathionine in presence of cystathionase enzyme. Cystathionase enzyme. It is hydrolytically cleaved. It is again a pyridoxal phosphate dependent reaction. PLP dependent reaction forming a molecule of water is being added, forming cysteine and homoserine. Forming cysteine. Forming cysteine and homoserine. NH2, COH, CH2, SH. Forming, that is cysteine. Cysteine and here one OH group get attached, forming homoserine. Understand? So this is the structure of homoserine. And this is cysteine. And we studied how from methionine, cysteine is being synthesized. From methionine will undergo activation forming S-adenosyl methionine, and a methyl group is removed forming S-adenosyl homocysteine. From S-adenosyl homocysteine, the adenosyl group is removed forming homocysteine, and this homocysteine will combine with serine in presence of cystathionine beta synthase enzyme forming cystathionine, and this cystathionine in presence of cystathionase enzyme, it is hydrolytically cleaved to give cysteine and homoserine, forming cysteine and homoserine. And this homoserine is then, by removing a molecule of ammonia, it forms alpha ketohydroxy butyrate. From alpha ketohydroxy butyrate, the carbon dioxide and water are getting removed, and a coenzyme A is being attached, forming propionyl CoA. Propionyl CoA is converted to succinyl CoA, and that is the catabolism of methionine. So, students, we can recap what we have studied today. Yes. What all we studied? Yes, we studied serine. Serine, what are the important points to remember in serine? Serine is an important 1-carbon donor. It's needed for the synthesis of choline. It is needed for the synthesis of glycine. And from serine, important phospholipid, glycolipid, and important proteins are getting synthesized. And also, serine is an important for the formation of the 21st amino acid, selenocysteine. Understand? And the selenocysteine, the important enzymes containing selenocysteine are deiodinase, thioredoxin reductase, and glutathione peroxidase. That is about serine. Now moving on to alanine. Important is In alanine, that the alanine, the Cahill cycle is important and beta alanine is important. Beta alanine is an important component of coenzyme A. Now moving on to methionine. Methionine, it is a sulfur-containing amino acid. It is an essential amino acid and it is glucogenic amino acid. An important role of methionine. What are the important role of methionine? It is involved in transmethylation reaction because of the production of the active form of methionine, that is S-adenosyl methionine. So it is important in transmethylation reaction. Second is that it is important for the synthesis of cysteine. From methionine, cysteine can be synthesized. And also methionine, you all know it is an initiator amino acid. It is an initiator amino acid in protein synthesis. So these are the important points to remember in methionine. Methionine is an important donor of methyl group as the active form of methionine, that is S-adenosyl methionine, and it is an initiator amino acid and it is needed for the synthesis of cysteine. How cysteine is getting synthesized from methionine? Methionine is activated to S-adenosyl methionine to S-adenosyl homocysteine, and this S-adenosyl homocysteine, by removing the adenosyl group, it forms homocysteine, and this homocysteine will combine with homocysteine. How homocysteine coming to the synthesis of cysteine? Please do students respond. S homocysteine will combine with serine. The enzyme is cystathionine beta synthase. It is PLP dependent enzyme forming cystathionine, and this cystathionine again by the action of cystathionase enzyme removing a molecule of water, again it is PLP dependent enzyme forming cystathionase enzyme forming cysteine and homoserine. And this what happens to the homoserine? Homoserine. What happens to homoserine?
Converted to propenyl CoA and succinyl CoA.
Yes, homocysteine, by removing a molecule of ammonia. By removing a molecule of ammonia, it is converted to alpha hydroxypyruvate. And this alpha ketobutyrate, alpha hydroxy ketobutyrate. And this alpha ketobutyrate, by removing a molecule of carbon dioxide and ammonia, it forms, and attaching a coenzyme A, it forms propenyl CoA, and propenyl CoA to methyl malonyl CoA, and finally to succinyl CoA. So that is the catabolism of methionine. And since succinyl CoA is getting formed, it is glucogenic. Succinyl CoA can enter into glucogenic pathway. Thank you, students. Yes, I think it's clear to you. Yes, please do respond. Clear,
Yes,
Ayo?
ma'am.
Yes, ma'am.
Are you able to take down the notes?
Yes, ma'am.
Yes. Say you take down the notes. I won't be using the PowerPoints. PowerPoint veno? Eh? No. No, I don't like to use PowerPoint. And PowerPoint ഉണ്ട്, but I don't like to use PowerPoint. That's why. I think it's clear to you. Very important. Homocystinuria. Next class we will deal with cysteine metabolism and the disorders associated with sulfur-containing amino acid. Thank you, students. Thank you very much.
Thank you.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.